All right, good morning, everybody. Thank you for coming on the last day before break. Before we get started, I just wanted to tell you, I'm almost done grading your exams. I'm probably about 2/3 done. I'm determined to finish before I leave today, because like I said before, I don't want homework either. So I'm gonna finish getting everything graded, hopefully by a couple hours after class is over. And then I'll send you an email and let you know how it went. But hopefully your grades will be up by today sometime, so keep an eye out for them. Does anybody have any questions about the exam or anything like that? I'm not going to tell you answers or anything. We'll talk about that after. I still have a couple people who haven't taken it. All right. Okay, so we're going to get started. We are moving into module number 2, and the kind of general gist of module number 2. So module number 1 was just kind of an introduction. We looked at what makes up a cell, the physical properties of individual types of cells and things like that. The overall gist of module 2 is how bacteria grow and survive. So how they eat, how they metabolize, how they defend themselves, all of those things is going to be what we're going to talk about in module 2. And then just to kind of go over what we're going to talk about later, module 3 is how we as humans control bacteria growing. So module 2, we're talking about how bacteria grow, what food they eat, what temperatures they need, what types of environments they like. And then in module 3, we're going to take that same information and we're going to say, okay, how do we as humans take advantage of that to kill microbes and to kill bacteria and to prevent them from making us sick? And then all of module 4 is going to be talking about diseases and what happens when bacteria succeed in making us sick, which is always my favorite part. It's not everybody else's favorite part, but I think it's the most interesting. It's most related to humans, so a lot of people tend to react to that one the most and like it the best. But all right, so we are moving on to chapter 6. And I will be posting a recording of this lecture if you miss it today or if you just want a refresher when we get back. I know I hate the winter break where we have one lecture and then you're gone for 3 weeks and then you have to remember it. But there will be a recording there. Rewatch it if you forget. So we're moving on to chapter 6 today. It's microbial nutrition and growth. So we're going to look at basically how microbes eat and what they eat. Because just like every other organism on the planet, microbes require some type of food to survive. Now, how microbes eat and what they eat are very different than what we think of as eating, right? We as humans think of eating as chewing food and swallowing it and having it go through our digestive tract. That is eating for us. Single-celled organisms like bacteria and archaea and fungi and things like that do eat. They just eat differently than we think that they do, or than we do. So just like all other organisms, they do require some type of food. But something to remember is that planet Earth has a nearly infinite number of different environments. You just can't even think of all the different environments, especially when you're thinking of something as tiny as a bacteria. A bacteria can live in any number of teeny tiny microenvironments, from being a bacteria that prefers to grow in the armpits of humans to bacteria that like to grow underneath rocks in soil, right? There's all of these teeny tiny microenvironments. And with every single microenvironment, there is an organism that likes to live there. Okay, so organisms, bacteria, and all of these teeny tiny organisms, there are infinite numbers of ways that they can get food. They all eat different things. They get their food in different ways. And so we're not going to spend a whole lot of time talking about individual bacteria and what their food is like. But we are going to talk about general groups of organisms. Just like everything else, we like to group things as best we can, put things into boxes. And the 2 boxes that we're going to talk about for bacteria and other microbes and what they eat are 2 things. So all organisms, regardless of domain or genus or species, require 2 things in order to survive. And these are two things that they have to acquire from somewhere. One of them is a source of carbon. Now, carbon is an element that is the building block Yeah. Oh, sorry, guys. Thank you for telling me. I'll come up eventually. Like that? Maybe. Oh, come on. Don't do this to me today. Aha. There we go. So all organisms require a source of carbon, and carbon is an element that is the building block of all organic molecules. Okay, so all of those macromolecules that we talked about, DNA, proteins, lipids, carbohydrates, the base element and the most common element in all of those molecules is carbon. And in order to survive, the bacteria have to have a way to build these molecules, or the organism, every organism, has to have a way of building these molecules. Without the carbon, they cannot build macromolecules. So they have to have a source of carbon. And then the second thing that every organism on this planet needs to survive is a source of energy. And the energy allows them to build those macromolecules. We talked very briefly about adenosine triphosphate, also known as ATP. ATP is the energy source for most organisms. That's what they use to build those macromolecules. It's how cells take those individual carbon molecules or those individual glucose molecules or those individual nucleic acids and put them all together to form these big, useful compounds. Okay, they require energy. All right, and it's the same thing as to go back to our Lego tower analogy. In order to build our Lego tower, we need the building blocks, which would be the carbon. So we need the pieces, and we need the energy to put them on top of each other, right? That takes physical effort. Not very much, but it takes physical effort to build a Lego tower. So we need the pieces, and we need the energy to do it. And at the base of all metabolism and life and everything that cells do in order to be alive, to grow and reproduce and metabolize and eat, they need these two things, energy and carbon. And this applies to humans as well. It applies to animals, plants, bacteria, everything. Okay. So first we're going to look at, and we're going to give names to the organisms that belong to these certain categories. So first we're going to look at where organisms tend to get their energy. And there's two groups, right? Everything's going to be broken down into two groups. I'm going to say that about 100 more times. Okay? So there are two general groups of organisms, or two general ways that organisms get their energy. And it's easiest for this one if you look at the name. Again, remember, if you look at the prefix or the suffix in the word, a lot of times that tells you what it means. Okay, so a phototroph, photo as a prefix, like photography or photosynthesis or any of those photo words, it just means light. Photo just means light. So organisms that get their energy or their ATP, right? Remember, we're using ATP as energy. Organisms that use electromagnetic radiation in light. So they use the energy produced by light waves to make their own ATP. Okay, those are called phototrophs. And this is like a good example of a phototroph is a plant. Okay, a plant sits out in the sun, it stretches out its green leaves, and those green leaves soak up the electromagnetic radiation and produce ATP. Now that process is fantastically complicated, and if you ever talk about photosynthesis and chloroplasts and things like that, you'll see how that actually works. In this class, we're not really talking about that. Okay, it's a lot of biochemistry, and it's very, very, very complicated. But all you have to remember is that phototrophs take light energy and turn it into ATP, which is like chemical energy. It's energy that cells can use. Okay, so these are things like plants and algae. Organisms that do photosynthesis. There are some amount of bacteria that can do this as well, but not very many. Okay? And then the other group are the chemotrophs, and chemotrophs use chemicals to produce ATP. There are lots of different chemicals that organisms can use to produce ATP. By far, the most common one are sugars. So animals, plants, bacteria, most organisms, if we had to go by the numbers, use some type of sugar to produce their energy. Humans most commonly use glucose, though we can also use lactose, like from milk, and fructose from like fruits. We most commonly use sugars. Plants also use sugars from soil. Don't take that out. Pretend I didn't say that. That was wrong. Animals most commonly use, sorry, I was out late last night. Animals most commonly use sugars. You can also use things, there's certain bacteria that can use things like ammonia. There are certain bacteria that can use metals like iron and mercury to produce ATP. But all of those things are chemicals. And by far the most common one we're going to talk about are different types of sugars. So that's the big one. But chemotrophs in general use chemicals to produce ATP. So light or chemicals. Photo, chemo. Okay? Now the second thing is a source of carbon, where that organism gets its carbon from. Now again, with the prefixes, so an auto, if you look at the prefixes of these words, autotrophs and phototrophs, autotrophs and heterotrophs, auto as a prefix means self. Like an autobiography is something that you wrote yourself by yourself. Okay, so auto is something you do, it means self. Hetero means other, or different, or opposite. Okay. So an autotroph is an organism that produces their own carbon. They are capable of making their own carbon, and the way that they're able to do this is they pull gaseous carbon dioxide. So carbon dioxide is what we call inorganic. It's a gas. It's floating in the air. Okay, but they can pull the carbon straight out of the atmosphere and form organic carbon from this inorganic substance. Okay, this is also what plants do, right? Plants get their carbon by taking carbon dioxide out of the atmosphere, pulling the carbon out and using it to make their own macromolecules, and then expelling the oxygen back out into the atmosphere, because that's what's left, right? If you take CO2, that's a carbon and two oxygens. If you pull off the carbon, what you're left with is oxygen. And that is what plants and algae and things like that put back into the atmosphere to provide the rest of us with oxygen. That's how all of us are able to survive, because there were organisms that pulled the carbon out of the carbon dioxide and made oxygen. So that is something that they are able to do. They do it within their own cells. They're able to create their own organic carbon. Okay, so that's why they're autotrophs, because they do it themselves. They create it themselves. Heterotrophs obtain carbon by consuming other organisms. So this is going to be like most bacteria and animals. Most fungi do this too. Most organisms do this. Okay. So we kind of think of the autotrophs of the world as the producers, right? They're the ones that actually produce the carbon. They make it themselves by pulling it out of the atmosphere. And then the heterotrophs are the consumers. They're the ones that eat the organisms that produce the carbon. So things like grass and plants, some bacteria, some algae, pull the carbon out of the atmosphere. And then the rest of us, the animals, the other bacteria, the funguses, we eat the plants and animals that produce this carbon. Okay, so hetero means you got it from somewhere else. You're eating it from somewhere else, taking it from something else. All right. So when you combine these two categories, right, where you get your carbon and where you get your energy, you come up with 4 individual categories, right? If you look at the energy, remember energy, we have chemo or photo. And then if you add on the carbon source, you get these 4 categories. Chemo autotrophs get their energy from chemicals, right? This first word is where they get their energy from. So this is the first word, where they get their energy from. And the second word, or the second part of the word, I guess, is where they get their carbon. So if they get their energy from chemicals and their carbon, they make it themselves, they're auto, they make it themselves from CO2, they are chemoautotrophs. You just combine the 2 words. Okay. If they get their energy from chemicals, but they have to eat organic carbon, meaning they get it from somewhere else, they have to eat other organisms that have produced this organic carbon, then they are chemoheterotrophs, right? And then the same thing goes for the light ones. So if they get their energy from light, but they produce their own carbon from carbon dioxide, they're photoautotrophs. And if they get their energy from light, but they have to consume organic carbon from another organism, they are photoheterotrophs. So we have 4 different categories. Now, you don't necessarily have to memorize these words together. Like if I ask you what a photoheterotroph is, you don't necessarily have to memorize those words together, but if you memorize what this means, you'll be able to figure out what this means. Okay. Now, the 4 categories that we have here, 2 of them are relatively unusual, okay? And we're not going to talk about them much. And those are the ones on the edge. So chemoautotrophs and photoheterotrophs are kind of the outliers. There's a couple of bacteria that do this kind of metabolism, but it's very uncommon. Our 2 biggest categories in the natural world are chemoheterotrophs and photoautotrophs. Now, photoautotrophs, like we have here, are mostly plants and algae. Okay, so we just talked about plants. They're photosynthetic. They get energy and nutrients, energy and carbon from the atmosphere. They get light from the sun, and they pull carbon dioxide out of the atmosphere, and that's how they survive. Okay, algae do similar things. The other group, the chemoheterotrophs, are like animals, right? Animals are chemoheterotrophs. I can't go lay out in the sun and not have to eat for 2 weeks, right? That's not how it works. I have to consume my energy from other organisms. I eat plants and animals to get both my energy and my carbon. So animals, funguses, and most pathogenic organisms. Most pathogenic bacteria are also chemoheterotrophs. And that should kind of make sense. If you think about, when we're talking about pathogenic organisms, we're talking about bacteria or organisms that make humans sick. They make us sick because they're living inside of our bodies, right? Do you think the inside of a human lung is a great place to get sunlight? No, it's not, okay? They're living inside of our body because they're consuming us. They are eating the things that are inside of our body. They are taking energy and carbon from our cells. That's why they're living in there, and that's why they cause us to be sick. So it should kind of make sense that they're chemoheterotrophs because they have to get their energy from us. Now, there are lots of bacteria that are not chemoheterotrophs, but they don't tend to be pathogens. Okay? There are organisms that live in the ocean or in lakes and stuff like that, and that's where they get their energy from. They don't have to bother with us. They don't need us. Okay, so most of the organisms that we're going to be talking about in this class are going to be chemoheterotrophs. And we talked about that briefly when we talked about fungi at the end of chapter 4. And we talked about saprobes and parasites and the differences between those. So this is just kind of revisiting that little bit. But those are the 4 categories of nutritional diversity. There are 4 general ways that organisms get their energy and carbon and how they survive as organisms. Okay? And those are the 4 categories. All right, so now that we've looked at the kind of what they eat, so what organisms eat, how they or what they're looking for. Now we're going to talk about how microbes eat. So again, when we talk about eating, the thing that comes to mind for us, which makes sense because this is all we've ever done, is the idea of putting food in our mouths and chewing and swallowing and the digestive tract and things like that. Microbes don't eat the same way. Remember that bacteria and most microbes are unicellular, which means they're made up of a single cell. They don't have a digestive tract. They don't have a mouth. They don't have a stomach. They don't have an anus to expel waste, okay? They are one cell big. So the idea of what eating is something completely different in bacteria. Eating in bacteria is mostly just taking stuff from the outside of the cell and bringing it to the inside, and that's it. But the big kind of barrier there is the cell membrane. So we talked kind of briefly in the first chapter, I think, about cell membranes and how cell membranes are what we call semi-permeable. Which means that some things can get in and some things can't. So since some things can get into a bacterial cell and some things can't, how does a bacteria bring in all of the things that it wants to eat, all of the things that it needs to consume? How do those things get across the cell membrane? And that's what we're going to talk about now. So first we're going to talk about simple diffusion. And hopefully this isn't a brand new idea. You've probably covered this at least once in high school in some kind of physics or chemistry or biology class. It's the idea of diffusion. And simple diffusion is the movement of molecules from high concentration to low concentration. And that's the big thing. And this is what will naturally happen in any kind of chemical system. Now, this is due to a lot of things that we're not going to talk about. There are a lot of forces and, nucleus vibrating and electrons and valence electrons and all kinds of different stuff that we're not going to talk about. For now, I just kind of want you to take this at face value that in the natural world, in a system, things will always naturally go to a place where everything is distributed equally. And a really simple example of that is in this experiment right here. If you were to take a cup of water, it doesn't have to be a beaker, just whatever, and you can go home and do this if you've never done it before. If you go home and you take a big glass of water or a bowl of water, it doesn't matter, and you put in 3 drops of red food coloring. Okay, when you first put in those drops are going to drop into the water and they're going to kind of hang out in one spot, right? Because that's where you dropped them. But if you leave it alone, you don't stir it, you don't shake it, you don't jiggle it, you don't do anything to it. If you leave it alone and come back in 20 or so minutes, all of those dye molecules will have distributed throughout the water and everything will be equal again. Okay? You don't have to stir it. This is just the natural movement of molecules. It is the natural state of being of the natural world that everything wants to be equal. And everything will equalize if you leave it alone for long enough. Okay? I wondered if I had a better example. Okay? And in order for that to happen, what the movement is always going to be from high concentration to low concentration. Okay, so where there is the most of something to where there is the least of something. So in our example here, the dye molecules in this first part, there are more dye molecules in this one spot than there are anywhere else. So what's going to happen is those dye molecules will move out to where there is less dye. Okay, and they will continuously move until there's equal amounts of dye molecules distributed amongst the water. Okay, this will happen if you put, like if you take a cup of water and you put salt in the bottom, and if you don't stir it, eventually the salt will dissolve and redistribute itself so there's equal amounts of salt in the water. Okay? Everything wants to be equalized. And that's kind of the big idea here. Everything is going to move from high to low naturally. And because that is the kind of natural state of being, there is no energy required to make this happen, right? Again, with the bucket of water, or the bottle of water. You don't have to stir it up or shake it. You don't have to put in outside energy to make the dye molecules distribute. They will on their own. There is no energy required. It is just how things settle. Okay? And this will also happen across a membrane if the only way that this will happen is if molecules can cross that membrane. Remember, we talked about last time, or a couple chapters ago, that there are very few things that can cross a plasma membrane. That's why it's called semi-permeable, because some things can cross and some things can't. Can anybody tell me what can cross a membrane freely? Water. And I think I heard it, electrolytes. Some of them. There are very few salts, electrolytes, that can cross. Not all of them can. There are a couple, mostly sodium and potassium, but they can cross. Everything else is too big and will not cross a membrane. But if a molecule is capable of crossing a membrane, so say that this is a molecule of sodium, and it is capable of crossing a membrane, it will move across the membrane naturally from high concentration to low concentration. Okay, so you can see over here it's got 1, 2, 3, 4, 5, 6 molecules, and over here there's only 3. So these molecules will naturally flow across the plasma membrane to the inside of a cell. This is the inside. This is the outside. Will naturally flow inside the cell, what we call down its gradient, meaning from high concentration to low concentration. Flows downriver. Okay? And that is what diffusion is. But remember that this diffusion only works if molecules can cross the membrane. And there's only a couple things that can do that. It's mostly just water. There's a couple of electrolytes that can. It's mostly just water. All right? Now, osmosis is a pretty confusing concept the first time we talked about it. So I'm going to do my best to explain it. I do recommend there is a group that makes videos, and they make videos for all kinds of stuff, but they are called the Amoeba Sisters. And the Amoeba Sisters YouTube channel, they have tons and tons and tons of video that are generally pretty simple. They're about the level that we cover stuff, so they don't get too detailed, but they have moving diagrams, which I think it can be really helpful in things like this. Sometimes it's just, it's just better if you see it in motion, and sometimes that can help. So if after this you're still really confused, try their videos. They can be very helpful. So osmosis is it's a specific type of diffusion where it's the diffusion of water across a semi-permeable membrane. Okay, so osmosis only refers to the movement of water across a membrane, not any other molecules, not electrolytes, water only. And this particular scenario happens a lot, okay? And that's because there are a lot of things that cannot cross a plasma membrane, right? Water and a couple of electrolytes, but most things cannot cross a plasma membrane. So in this particular scenario, we're pretending that this is a cell, right? It doesn't matter which side is the outside, which side is the inside, but we'll say that this is inside and this is outside, okay? And this is inside and this is outside. So say a cell gets put in this scenario right here, where the inside of the cell has, if we look at it from what's happening with the solutes. Now the solutes are the stuff in the water, okay? So it's these little purple dots. These are solutes, and that can be anything, but it just basically means whatever stuff is floating in the water. That can be proteins or sugars or other cells or anything. Full salts like sodium chloride, it can be anything. It just means stuff. But if we're looking at this in terms of solutes, the inside of this cell has less solutes than the outside, right? So if you look at the picture, the inside has what, 1, 2, 3, 4, 5, 6, 7 things floating in it. The outside has more than 7, very clearly, okay? Now, the natural world wants this to be equal. But the problem is that because we have added this semi-permeable membrane, the stuff can't move, all right? So this stuff, whatever it is, we'll say it's a protein. This protein can't move. It can't move. If the protein was moving, right, which way would it move? Would it move to the outside or to the inside if the protein could move? Which way do things move, remember? High to low. So if the protein could move, where is it higher? Outside or inside? Outside. There's more protein outside, right? So if the protein could move, it would move inside. But it can't. So because the protein can't move, what we want to look at is the concentration of the water. Now, and this is talking about how much water there is. So we're talking about concentration of the water. It is how much water relative to the stuff, relative to the solutes floating in it. Okay, so in this first scenario over on this one, my left, but I don't know what side it's on for you. I guess it's on the left for you too. So on the left for you too. And the inside of this fake cell, there is there's more water than solutes, right? There's more water than stuff. Like the water is the pink stuff, and the stuff inside of it is the purple bally things. There is more pink liquid than there are purple bally things, right? There's more water than stuff. And on the outside of the cell, you have less water than stuff. Okay, there's about equal amounts of pink liquid to purple stuff. So what is going to happen is, because the only thing that can move in this scenario is the water. So where do you have more water? We just said it. Do you have more water inside or more water outside? You have more water inside. So the water is going to move from high to low, just like we just said. So the water is going to move because the water is the only thing capable of moving here. And so the water will move to balance out the amounts, the relative amount of water to solution, to solutes, to stuff floating in the water. So in the scenario on the right, after diffusion has happened, there are equal amounts of pink liquid to purple bally things. Okay. And so this scenario happens a lot, and we're going to talk about tonicity in just a second. That has to do with osmosis. And this is what happens across plasma membranes. You have water moving into cells and water moving out of cells constantly to keep things balanced, because we're going to talk about what happens if they're not. All right. So the water will move to whatever side it has to balance out the amount of water relative to the solutes there. All right, so what tonicity is it has to do with the amount of water and the solution when you're looking at a cell, okay? So it has to do with the solution outside of a cell and what happens to that cell in a particular solution. So I guess, yeah, the first one we're going to look at is an isotonic solution. Okay, an isotonic solution. And what an isotonic solution means is that the water concentration is equal inside and outside the cell. Okay. So the amount of water relative to the solutes is equal inside and outside the cell. That means that this cell is going to be really happy. Water may flow in a little bit, water may flow out a little bit, but everything is going to stay relatively equal. Everything is kind of at equilibrium. Okay, this is a happy cell. The water concentration is relatively equal inside and outside, and everything remains in equilibrium, and nothing is going to happen to that cell. And we're going to look at what happens with cells with cell walls and cells without cell walls. But in both of these scenarios for an isotonic solution, those are happy cells, okay? There's no net water movement. Everything is just equal. Everything is in equilibrium, okay? Now, in a hypotonic solution, hypo as a prefix means low. Hypo is low. Okay? Like hypoglycemia is when you have low blood sugar. Hypo is always going to mean low. So in a hypotonic solution, and the word always refers to the concentration outside the cell. Okay, so hypotonic means that there are less solutes outside. Okay, so outside the cell is low on solutes. Okay. So remember, and it's just like this scenario right here. So if there are lower numbers of solutes, that means that the amount of water is higher. I know it's a little backwards, But if you're looking at it relative to the inside, so outside the cell, we have two solutes. And the rest of this is water. I know it's not pink here, but imagine that it's pink all around the outside. Okay, inside the cell, we have a lot more than 2 solutes, right? And so that means that we have less water. Okay, so in this case, we have fewer solutes, low solutes outside, which means you have a high amount of water. So water is the only thing that can move here. So the water is going to move inside the cell. Okay. So the net diffusion of water is going to be into the cell. And this can swell the cell. It can make it get bigger. See here, this is a normal cell over here. This is what a cell looks like when it's, happy. And this is what it looks like when water is flowing into a cell. And in a cell without a cell wall, like a human cell, this can cause these cells to burst open and die. Okay. And so there are cases of this. If you've ever heard of this, this can happen in humans. Okay, this can happen within a human body. It's called water poisoning, and it can happen. I think it happened. I don't know if it was this summer or last summer. It was kind of a big TikTok thing, but there was a woman who went to the beach, and this just went around TikTok. I don't know the details of it, but she went to the beach. She had spent all day outside. She was hot. It was sweaty. And she hadn't been drinking any water. So she was dehydrated. She went home and she was really dehydrated. And so when she got home, she chugged a literal 8 bottles of water in 5 minutes. Okay, so what that did was that made her blood too watery. It made her blood an isotonic solution, because it diluted out everything that was in her blood. There was too much water on the outside of her cells. And this is exactly what happened to her red blood cells. Because she consumed so much water so quickly, her red blood cells, all the water moved into the red blood cells to try to balance out those concentrations, and her red blood cells burst and she died. It's a very real thing that can happen. If you are severely dehydrated and drink too much water too fast, you can die from that. It's called water poisoning. It's usually much less dramatic than that, but this is what can happen. Okay, that's like the extreme version of an isotonic solution. And the other thing that's kind of important to remember about all of this is that as humans, we are a full system, right? I have multiple millions of cells in my body, and I have an organ set called kidneys that deal with balancing out the solution that is my blood. Okay, that's what your kidneys do. Your kidneys spend their whole day filtering your blood and keeping your blood at an isotonic solution. That's their whole job. That's why if you're drinking more water than normal, your kidneys flush out more water. Your kidneys are trying to keep everything balanced. That's their big job. But bacteria and other microbes are mostly unicellular. What that means is they're just floating around in whatever's around them. They don't have kidneys to balance out their environment. They're just floating in whatever happens to be around them. And so if they happen to be in a hypotonic solution like this one, where there's not enough solutes, there's too much water, the water will flow into the cells to try to balance it out, and those cells get bigger, they swell up, and they can eventually burst and die. Now the opposite is called a hypertonic solution. Now hyper, like a kid who's had too much sugar, is hyper. Hyper means high. And what you have here is exactly the opposite of a hypotonic. So this is the solution outside the cell has too many solutes. It's a high solute concentration, which means there is less water. And so what will happen is the water will flow from the outside of the cell, from the inside of the cell to the outside, which will cause the cell to shrink up. And it can eventually die as well and become distorted. A really great example of this, again, in humans, has anybody ever spent, maybe not in a while, but have you ever spent like the all day in the ocean and you come home and you feel like your skin is too tight and it's like drying off and you feel like you can't drink enough water? It's because you spent the whole day submerged in a hypertonic solution. And all of the water is being pulled from your body to try to balance out those concentrations, okay? And so that's why your skin feels incredibly dry. It feels like it's too tight, like there's just no water left. Your lips feel dry. That's part of why like you get wrinkly when you're sitting in especially salt water for a really long time. And you feel like you could just like sit in a bath and soak up the water because your body is so dry. That is because you have been sitting in a hypertonic solution. So because salt water has lots and lots of salt in it, it has lots and lots of stuff, has lots and lots of solutes, okay? So that is a hypertonic solution. And what happens again in a hypertonic solution is the water flows out of the cell to try to balance out those concentrations. It means that there's too much water, or there's not enough water outside. We'll say it that way. There's not enough water outside, so the water moves out to try to balance that out, and the cell will shrink up, like sitting in the ocean all day. Your skin feels like it's shrunken. All right? So that is tonicity, and that is a thing that be it bacteria have to deal with on a regular basis, because again, remember that they're just floating around. And kind of an interesting way to think about this, and we're going to talk more about it in Module 3, we'll bring it up again, is that one of the ways that we control bacterial growth, before we had refrigerators or before we had freezers and stuff like that, how did people preserve meat? A lot of times. They either dried it, so they sucked all the water out, or they made like salt pork or bacon, things like that, where you're drenching it in salt. And that was to prevent bacteria from growing, because bacteria cannot really grow in hypo or hypertonic solutions. Things like jam and honey and jelly, those all have tons of sugar, which is another solute. So honey is a hypertonic solution. Honey is one of those foods that basically never goes bad. And it's because it's extremely hypertonic. Bacteria basically can't grow in it because there's too much sugar in it. It's a hypertonic solution. All right, so that was osmosis. That was dealing with how water specifically moves across a plasma membrane. Now, other ways that, because bacteria and cells and microbes have to eat things other than water, right? So they have to get things across their membranes. So how do they do it if things are not going to cross the membranes? And there's a couple of different ways. So the first one is facilitated diffusion. What facilitated diffusion is just like the diffusion that we talked about over here, where things moved from high concentration to low concentration. But this is a molecule, like we'll say this is a sugar. And remember that sugars and proteins and pretty much everything else are too big to cross the plasma membrane on their own. So they won't be able to cross the plasma membrane. But if a cell wants to be able to bring that molecule inside of its cell, it will have a specific channel in the plasma membrane that is specifically meant for that molecule. And there's usually one channel for one type of protein or sugar or other molecule. They're very specific. So if a bacteria frequently, its major food source is lactose, it's going to have a lactose channel, because that is what it uses as its food source. If there's a bacteria that likes to consume, there's bacteria that can consume iron, right? It's a heavy metal. They'll have iron channels, okay? They'll have channels very specific for whatever it is that they want to consume. And different bacteria and different organisms and different cells consume different things. And so they will have different channels. Now, the way facilitated diffusion works is that there is no energy required for this because the solute is still moving from high concentration to low concentration, right? Here we still have, there's 1, 2, 3, 4, 5, 6 molecules outside and 1, 2, 3 inside. So if that channel is available, it will cross. It will cross over. And again, there's no energy required because it's still moving down its natural gradient. It's moving from high concentration to low concentration. It is facilitated diffusion. It's just doing it without any energy, but it just needs a little help. It's facilitated. It needs a little helping hand. It's like crossing a river, right? I can't cross a river. I guess I could, but I can't cross a river unless there's a bridge. It's like a bridge. The channel is like a bridge. It's just a place that allows them to cross over. Next, we have active transport. Now, active transport is specifically moving, oh, I have it written down already. The active movement of solutes across a semipermeable membrane, specifically against the gradient. So organisms will move, or solutes will move naturally from high concentration to low concentration. But if an organism specifically wants to collect all of a specific solute, it can do active transport to pull the solutes away from their natural gradient. Okay? And to do this requires energy input because it's doing something against the natural equilibrium, right? The natural equilibrium is to move from high concentration to low concentration. But again, if an organism is choosing to eat lactose, right? The lactose is their chosen food source. They want all the lactose they can get. Well, if you have, in this case here, like think about this facilitated diffusion, if all things remained the same, right, a couple more of these would move in, and then we'd have equal amounts, right? And then we wouldn't be able to get any more because it's not going to move across its gradient. But say that the organism still needs more lactose. I need more. I want to eat all of it. It can pull it actively by using energy and a special type of carrier protein to make that happen. We're going to talk more about using ATP later, so just kind of take this at face value for now. But it basically uses an ATP. It uses an ATP as energy to pull something away from its natural gradient. So naturally, we have low concentration here and high concentration here. So if we allowed this to happen across its natural equilibrium gradient, this solute, whatever it is, would move from high concentration to low concentration. It would flow back out of the cell. It would go this way. Okay, but that's not what we want to happen here. This bacteria or this cell wants all of this solute inside. So it's going to use this active carrier protein and some energy to keep moving these solutes inside the cell, even though that's against its natural gradient. And that's why it requires energy, because it's against equilibrium. Okay. And if you ever take anatomy and physiology, or if you have already, this is basically the basis of how your entire nervous system works. So your entire nervous system works because of something called sodium-potassium pumps, and it pumps sodium and potassium across its gradient, and that's what creates action potentials, which allows your entire nervous system to move. You don't have to know that for this class, but just make a connection. If you're nursing majors, you'll have to take anatomy and physiology eventually, or you have already. So active transport is incredibly important, but you just have to remember that it does require energy input. right? Okay, so now that we've talked about what it is that microbes like to eat and how they eat it, so how they get those molecules inside their cells, we're going to talk about some of the other factors that go into microbes being able to survive, and that is the environmental factors. Okay, and mostly when we're talking about environmental factors, we're going to be talking about temperature, the gaseous atmosphere, so mostly how much oxygen there is. We'll talk a little bit about pH, which I'll explain in a minute if you don't know what that is. I think that's it. Okay, we're going to go through each of these individually. All right, so the first one we're going to talk about is temperature. So again, just like every other organism on the planet, bacteria have a specific temperature range at which they can grow. And different species of bacteria have different temperature requirements. We all have, animals and stuff have different temperature requirements, right? Like I couldn't live in the same places that polar bears live. I wouldn't survive there. Polar bears are naturally designed to grow in much colder temperatures than I am. I'm not covered in fur. I would die out in the Arctic, right? So animals and plants and things like that are all specifically suited to particular environments, and bacteria are exactly the same, okay? So we are going to talk about the different categories of temperature and the different categories at which bacteria like to grow. And yes, you have to know all their names, and yes, you have to memorize the temperature ranges, okay? So we have about 5 different categories. And when we're looking at these temperature ranges, there's going to be 3 different things that we're going to look at. The first one is the minimum temperature at which something can grow, the maximum temperature at which something can grow. So the minimum is going to be this blue dot on any of these. Okay. And then the maximum is going to be the pink dot over here. And those are the outside edges of how something can survive, right? And again, it's humans, and I'm going to talk about this in terms of like an individual human, right? So the maximum temperature, I actually don't know what the maximum temperature of a human is if you threw them outside. Like if they didn't have any air conditioning, it's probably what, 100 degrees? Maybe a little less Fahrenheit. If you were just outside, you had no shelter, what temperature could a human survive at? Maybe 100, I don't know. That might even be a little too high. And then a human would have a minimum temperature too. A minimum temperature would be probably just above freezing. We couldn't survive below freezing. Maybe, 40 degrees Fahrenheit outside with no shelter, right? So there's a maximum temperature and a minimum temperature. But also think about if I was to be shoved outside in 100-degree heat without any shelter, am I going to be doing great? No. I might survive, but I'm not going to be doing great, right? I'm going to be barely surviving. And the same thing is true if you threw me outside at 35 degrees Celsius. I might survive, or 35 degrees Fahrenheit. I might survive, but I wouldn't be doing great. And the same is true for bacteria. They can survive at certain temperatures, but they may not do great. The third thing, which is the one that we're going to talk the most about, is the optimum temperature, which is the average temperature at which they grow best. And that's why this graph, it's showing it as a peak, because this graph is looking at the rate of growth. And so the rate of growth is going to be highest at the optimum temperature, right? My optimum temperature is 72 degrees Fahrenheit. My husband thinks his is 65, right? So each person has a different optimum temperature, and each bacteria is going to have a different optimum temperature. So we're going to focus mostly on the optimum when we're looking at these groups, but just know that the optimum doesn't mean that if it gets 1 degree above optimum or 1 degree below optimum, that it will die. It's just, that's the best, okay? So when we look at these categories, we're gonna start at the bottom. So the first one is a psychrophile. And there's the word right there, a psychrophile. And psychrophiles tend to grow at relatively cold temperatures. Psychrophiles, their optimum temperature is right at about like 4 to 5 degrees Celsius, we'll say. 4 to 5 degrees Celsius. Now, remember a couple of chapters ago, we talked about a couple of benchmarks for temperatures in Celsius. Remember that 0 degrees Celsius is freezing. 4 degrees Celsius is about refrigerator temperature. Okay? So the optimum temperature for psychrophiles is about 4 to 5 degrees Celsius. That means that their best temperature is in the refrigerator. But look at that, they can also grow down to well below freezing. That may not be their best temperature, but psychrophiles can grow in ice blocks. They can grow in glaciers. They can grow in Arctic oceans that are very close to freezing or below freezing. They can grow in those extremely cold environments. So we call psychrophiles like the cold to extreme cold. Again, for most of the organisms in this group, the average optimum temperature is about 5 degrees Celsius, which is about refrigerator temperature. But they can grow much colder than that. For the next one, we have psychrotolerant organisms. And psychrotolerant organisms are the next kind of temperature. And their average optimum is right about 25 degrees Celsius. So what is 25 degrees Celsius? What was the benchmark I gave you for that one? I think I heard it. It's about room temperature. So it's about 70-ish degrees Fahrenheit. It's about room temperature, an average room. Obviously, this room is colder than 70 degrees right now, but it's an average room temperature. It's about 25 degrees Celsius. So psychrotolerant organisms, their average optimum best growing temperature is going to be around regular temperature. So about in the environment, a lot of things that grow in just the soil and water and things like that are going to be psychrotolerant. Now, also keep in mind again, though, that these psychrotolerant, they can grow down to refrigerator temperatures, right? Their minimum is down to 4 or 5 degrees. So there are organisms that are capable of growing in the fridge, but also surviving at room temp, and even a little bit higher than that. But their average optimum temperature is about 25 degrees Celsius. Next, so these are, I kind of call these like average to cold, or like room temp to cold, if we had to classify them. Okay. Next, we have mesophiles. Now, mesophiles are their average temperature. It's right about, it's usually about 37 to 39 degrees Celsius. And what is 37 to 39 degrees Celsius that we talked about? The human body. The human body is at about 37 to 39 degrees Celsius, or about 98 degrees Fahrenheit. So these I kind of label as human to warm. Okay, so they like things a little bit warmer than average or warmer than room temp, but they don't like extreme heat. Okay. So next, I wish I had color-coded this more properly, but it is what it is. Okay, so next we have the thermophiles. And thermophiles, their average optimum temperature is usually about 70 degrees Celsius. Now, what temperature is boiling in Celsius? 100. I always think Celsius is easier because 0 is freezing, 100 is boiling. So 100 degrees is boiling. So this isn't quite boiling, but it's warmer than a human body. Okay, so it's somewhere in between a human body and boiling temperature. It's kind of warm. So I put these into warm, they like warm to hot environments. Okay, so these are types of bacteria that can live in like hot springs like in Arkansas. Those are usually just below boiling. They're not quite at boiling for the most part. They can survive in compost piles. I don't know if anybody's ever been near a compost pile, but they're weirdly hot. There's a lot of metabolism and decomposition going on in there, and they're very warm. So they can survive in compost piles, deep in the soil. It's very warm deep down in the soil. Places like that. Hot springs. On and on. And then our last one are going to be the extreme thermophiles. And their average optimum temperature is about 125 degrees Celsius, which is above boiling. So they are perfectly happy growing above boiling temperatures. So that is places like lava flows. There are thermal vents in the ocean where lava basically comes up from the ocean floor, and they're extremely, extremely, extremely hot. They can survive in there. So there are a very few subset of organisms that can survive in those extreme, extreme, extreme temperatures. There's not very many, but they do exist. Okay. So I put these as like extreme hot. Okay. I don't know why I want to redo this one. Okay. So this is cold to extreme cold. Okay, so those are the 5 categories of temperature for bacteria and the environments that they like to live in. So when we're talking about studying a microorganism, it doesn't have to be a bacteria, it's any kind of microorganism. We classify them into one of these 5 groups. Now, if you had to guess, what category do you think most human pathogens fall into? Most of them are mesophiles because they like to grow at human temperature. There are some very dangerous ones that are psychrotolerant because it has been true for many, since humans existed, we've been taking advantage of this to protect ourselves from bacteria, right? What the most basic form of bacterial control that we have done since before we knew that bacteria existed is what? Heating up our food. Right? We cook our food, we heat it up. And that kills almost everything, right? Boiling water, cooking your food is going to kill almost all bacteria because there are very few things that can survive a straight flame like that. And another thing that's come along more recently, but is just as important, is the idea of refrigeration and freezing, because there are very few organisms that can survive in the fridge. Or organisms that cause human disease that can survive in the fridge or the freezer. But there are a couple of examples of human disease, particularly things like listeria. Listeria is a big one. And listeria is a kind of switch hitter. It's really dangerous because it can grow in the fridge. It can survive the freezer, but it also grows well inside the human body. So it's one of these that kind of overlaps in here, where it can grow in the fridge, and it can grow in the human body. So listeria, if you've never heard of it, is a bacteria that is very common in like food spoilage. And every time there's been like a food recall, a lot of times it's listeria. It's also why pregnant people are not supposed to eat cheese and lunch meat and sushi and things like that, because listeria can grow in cheese because cheese is kept in the fridge. Especially not pasteurized cheese, and especially cheese that is being eaten raw. It's not uncommon to just eat cheese on crackers, right? You don't have to cook it. Remember, you guys might be a little young. I don't know how much longer I can tell this story. But do you guys remember when the Bluebell factory shut down? it was like 6 years ago, 7 years ago. So the Bluebell factory shut down. That was because they had a listeria outbreak. So one of the insides of their mixers was contaminated with listeria, and there was a huge outbreak of people getting listeria from ice cream. And they couldn't keep it in the fridge. Listeria can survive in the freezer, and then people would consume their ice cream and they got violently ill with food poisoning. So there are a couple of human pathogens that can span those, but the vast majority of human pathogens are going to be mesophiles. Okay? So next time we will talk about pH. We'll move on after the break. Everybody have a good break. I will see you back in January. And drive safe as you get home.